Hello, everyone. This is Dr. Cole, and this is the third part of our nervous system lectures. And today we're going to talk about synapses. So here's our typical neuron. And we see that, as we've learned before, there are the dendrites that receives information. And then that information passes through the neuron in the unidirectional manner. So there's a cell body, down the length of the axon, all the way to the axon terminals. And neurons are functionally connected to one another by synapses. So a synapse is defined as the junction between two neurons, or a neuron and a target cell. And so the neuron conducting impulses toward the synapse is the presynaptic neuron, whereas the neuron transmitting the electrical signal away from the synapse, that is the postsynaptic neuron. And at a given synapse, the presynaptic neuron sends the information and the postsynaptic neuron receives the information. And most neurons, as you'll see, function as both presynaptic as well as postsynaptic neurons. Okay. And neurons have anywhere from 1,000 to 10,000 axon terminals making synapses and are stimulated by an equal number of other neurons. Okay. And so if there was a neuron upstream of this neuron now becomes the postsynaptic neuron, okay, with relation to the synapse it makes with that neuron upstream of it. Okay. Now, outside of the central nervous system, in the peripheral nervous system, the postsynaptic cell may be either a neuron or an effector cell, such as a muscle or a gland. Okay, and here this shows an example of a neuromuscular junction. So it's a junction, or it's a synapse formed between a motor neuron and a skeletal muscle. So we talked about how information flow within a neuron is unidirectional. It enters the dendrites, moves through the cell body, down the axon to the axon terminal. Okay, and information flow in also one direction, only across chemical synapses. Okay. It flows from the presynaptic neuron across the synapse to the postsynaptic neuron. Okay. And the pattern you'll see is that electrical signal within a neuron gets converted to a chemical signal across the synapse. And then that chemical signal then gets reconverted back to an electrical one once it enters that postsynaptic neuron. One thing to keep in mind is that there's the synaptic delay, because chemical transmission is slower than electrical transmission. So that means the more synapses there are in a neural pathway, the more time generally it will take for that information to finally reach its destination. So every time that signal has to cross the synapse, there is a bit of a delay. Okay. So here is a zoomed in magnifying view of a synapse, and you see that there is a gap, okay, between the presynaptic neuron and the postsynaptic neuron. And that gap is called the synaptic cleft. So in other words, the presynaptic neuron and postsynaptic neuron are not physically in contact with one another. So these chemical synapses can be formed by different parts of a neuron. An axil dendritic is formed between axon terminal and dendrites. So axon terminal of one neuron and the dendrites of another neuron. Whereas axosomatic is between the axon terminal of one neuron and the cell body of another neuron. Axonic is between axon terminal of one and the axon of another. Dendritic is between dendrites of one and the dendrites of another. Okay. So here's a YouTube video that you should watch. You should pause this lecture recording and watch this video to get an overview of how a synapse works. Here we're going to take a look at detailed events happening at a chemical synapse. So last lecture, we talked about how action potentials are generated and propagated down the length of the axon. When this action potential reaches the axon terminal, okay, that depolarization of that axon terminal causes voltage-gated calcium channels in the axon terminal to open. Okay, and the opening of those voltage-gated calcium channels lead to an influx of calcium. So calcium enters into this axon terminal, and that causes these synaptic vesicles containing neurotransmitters to dock on that axon terminal membrane and fuses with the axon terminal membrane, and then release its contents, namely the neurotransmitters, into the synaptic cleft. From here on, the neurotransmitters then diffuse across the synaptic cleft and bind to the postsynaptic receptors. Okay, and because diffusion takes time, right, especially in comparison to the conduction rate of action potential. So this is why we mentioned a few minutes ago that the more synapses you have, the slower that information will get transmitted. Okay. In any case, the neurotransmitter binds to the postsynaptic cell, okay, and that binding opens up the ion channels on postsynaptic membrane, and that leads to an influx of positive ions, or it may be different ion channels that it opens. The result is a greater potential. Okay, and so how can we limit the effect of these neurotransmitters? Well, there are a number of ways. One of it is through enzymatic degradation. So there are enzymes waiting around in the synaptic cleft. Once that neurotransmitter has been used, or perhaps even before it's been used, it gets degraded. Okay, this effect can also be terminated by the diffusion of the neurotransmitter away from the synapse. And thirdly, it can also be terminated through reuptake mechanism back into the presynaptic neuron. Now, the higher the impulse frequency, meaning the higher the frequency of the action potential, the more vesicles will get exocytosed and leading to a greater effect on the postsynaptic cell. Okay, so this is how our nervous system is able to code for the strength of that signal. We're now going to move on to talking about postsynaptic potentials, EPSPs and IPSPs. So postsynaptic potential is a specific type of greater potential that occurs in the postsynaptic neuron. And this postsynaptic potential can vary in strength depending on the amount of neurotransmitter that's being released, as well as the amount of time that the neurotransmitter is kept in that synaptic cleft. And also depending on the effect of that specific chemical synapse, we can form either an excitatory or an inhibitory postsynaptic potential. Okay, so that's what EPSP and IPSP stand for. Let's now first look at the excitatory postsynaptic potential. So the neurotransmitter binding to the postsynaptic neuron can open up these chemically gated ion channels. Okay, and this is in contrast to the voltage-gated channels we see on axonal membrane. Okay, so at excitatory synapses, the neurotransmitter binds, depolarizes that postsynaptic membrane. Okay, and the ion channels that tends to open tends to allow both sodium as well as potassium to pass through simultaneously. Okay, and recall the chemical gradient of these two ions. One is going to enter into the cell, the other is going to exit out of the cell. So although this two-way cation flow may seem to be self-defeating when the goal is depolarization, But remember that the electrochemical gradient for sodium is much steeper than that for potassium. So as a result, the net effect is a sodium influx causing depolarization. So there's a net depolarization. Okay, and if neurotransmitter, if there are enough neurotransmitter that binds, the depolarization of the postsynaptic membrane can reach as high as 0 millivolts. That's way above the axon's threshold of about -50 for firing and action potential. Okay, so remember that unlike axons, which have voltage-gated channels that make action potential possible, the postsynaptic membranes generally do not generate action potential. And that dramatic polarity reversal that we see in axons pretty much never occurs on the membranes, in the postsynaptic membranes that contain only chemically gated ion channels because of that opposite movement of potassium and sodium, preventing the excessive positive charge from accumulating inside a cell. Okay. So for this reason, instead of action potentials, we see depolarizing graded potentials that we call excitatory postsynaptic potentials, EPSPs, occurring at excitatory postsynaptic membranes. So each EPSP lasts only a few milliseconds before the membrane then returns to its resting potential. The only function of EPSPs is to help trigger an active potential distally at the trigger zone of the postsynaptic neuron's axon. Okay, so although currents created by individual EPSPs will degrade with distance, they can and often do spread all the way to the axon's initial segment, to its trigger zone. And if that current reaching that trigger zone is strong enough to depolarize the axon to the threshold, then that postsynaptic neuron will fire an action potential through the action of those voltage-gated ion channels. So this next slide just has words summarizing what I just went over. In contrast to the EPSPs, the IPSPs, or inhibitory postsynaptic potentials, is the hyperpolarization of the postsynaptic membrane. It tells the neuron to not fire. Okay, so as the name indicates, binding of neurotransmitters at inhibitory synapses will reduce a postsynaptic neuron's ability to generate an action potential. Most inhibitory neurotransmitters hyperpolarize the postsynaptic membrane by making the membrane more permeable to either potassium or chloride. And so in these cases, the sodium ion permeability is not affected. And now consider if the potassium channels are to open. Potassium will move out of the cell, correct? Okay. So we've got positive charge moving out of the cell. The membrane will become more negative, right? If a chloride channel were to open, keep in mind that chloride concentration gradient is such that it's higher concentration outside the cell. Okay. So when we open chloride ion channels, chloride will move into the cell. Okay. And so we've got a negative charged ion moving into the cell. Okay. That's electrically the same effect as moving a positive charge out of the cell. Okay. So in either case, the charge on the inner surface of the membrane become more negative. And as the membrane potential increases and is driven farther away from the axon's threshold, the postsynaptic neuron becomes less and less likely to fire. And then the larger depolarizing currents are then required in order to bring about an action potential. Okay. So these hyperpolarizing changes in potential are called the inhibitory postsynaptic potentials. And once again, this is just a slide that summarizes what we just discussed. Okay, so summation of postsynaptic potentials. So one postsynaptic neuron can receive inputs, either as EPSPs or IPSPs, from thousands of other neurons. Okay. Thousands of other neurons. And it integrates these inputs at its initial segments in order to determine whether it will fire an action potential or not. In this illustration, we see there are about like 10 presynaptic neurons synapsing onto this one postsynaptic neuron. Okay. In reality, this number is more like thousands. Okay, 1,000 presynaptic neurons synapsing onto one neuron. Okay, and then this neuron can then be one of the many thousands that's synapsing onto another neuron. Okay, so it's a very complicated picture, but so how is this poor neuron supposed to decide whether or not to fire, right? Okay. So the summation of many EPSPs and IPSPs at the axon's initial segment will determine if a neuron reaches threshold to fire an action potential down the axon. And remember that the EPSPs and IPSPs are greater potentials that decay with time and distance as they spread down the dendrites and across the cell body to the initial segments. There, they can sum together, and an action potential will be fired if the initial segment or the trigger zone reaches threshold. Okay. So a single EPSP cannot bring about an action potential. But when we look at the summation of multiple and oftentimes thousands of EPSPs summating together, okay, that can have quite an influence on that postsynaptic cell. Okay, IPSPs can also summate, and oftentimes what we'll really see is a summation of EPSPs and IPSPs together. Okay, so there are two types of summation: temporal and spatial. So first, here's an example of EPSP without summation. Here in this image, you see that there are two stimuli that are separated in time. Okay, they're so separated in time that they do not add together. So now if you space these two stimuli closer together in time, it will cause the two stimuli to be summated together and help the axon reach the threshold needed to fire the action potential. Okay, so this temporal summation is referring to the excitatory stimuli close in time. Okay, and these excitatory stimuli generally come from the same presynaptic cell. Okay, and then we also have spatial summation. Spatial summation is stimuli happening at the same time, but at different locations. Okay, and those, when it gets transmitted through the cell's body, the postsynaptic cell's body, and then adds together at the initial segment. Okay, so we have a summation through space and summation through time. This slide summarizes what we just talked about in the previous slide. In reality, with that thousands of presynaptic neuron synapsing onto this one postsynaptic neuron, what we really see is a summation of EPSPs and IPSPs coming from these thousands of presynaptic neurons, okay? Or generated by the stimulus coming from the presynaptic neurons. And so the effect, the overall effect on the postsynaptic cell is kind of like a tug of war between the inhibitory PSP and the excitatory PSP. Okay. And so you can see that through the spatial summation of these two, they can actually cancel one another out. Okay, but now if the excitatory postsynaptic potential predominate. If we have more of that than the inhibitory postsynaptic potential, then the excitatory one will dominate, and it will help the axon reach the threshold needed to fire an action potential. The examples of synaptic integration we have discussed so far all took place on the postsynaptic side of the synapse. For example, we've taken a look at how two excitatory postsynaptic potentials can summate temporally and spatially. However, the activity of presynaptic cells can also be altered or modulated. When a modulatory neuron terminates on a presynaptic cell, the EPSP or IPSP created by the modulatory neuron can alter the action potential reaching the axon terminals of the presynaptic cell. Okay. And it can then modulate its neurotransmitter release. So in this image, we can see this modulatory neuron A, synapsing onto the terminal bouton of the presynaptic neuron B. Okay. Depending on the nature of this axo-axonic synapse between A and B, neuron A may either work to increase or decrease the effect that neuron B has on neuron C. Okay. And when neuron A increases neuron B's effect on neuron C, this is called presynaptic facilitation. Whereas if neuron A decreases neuron B's effect on neuron C, this is what we call presynaptic inhibition. As it turns out, both presynaptic facilitation as well as presynaptic inhibition work through activation or inactivation of calcium channels at the axon terminal. Okay, so let's take a closer look at the mechanism of action. Presynaptic facilitation can be elicited by neurotransmitter serotonin through its enhancement on the activation of calcium channel. So remember those voltage-gated calcium channels that open upon being triggered by action potential arriving at the axon terminal? Well, a neurotransmitter like serotonin can actually enhance it and causes more of those calcium channels to open. And more calcium influx means more neurotransmitter release, and therefore more neurotransmitter binding to the postsynaptic cell, and therefore increased effect on that postsynaptic membrane. Similarly, presynaptic inhibition can be elicited by a separate neurotransmitter called GABA. As you can see here, GABA can act to inactivate the calcium channels. Okay, so less calcium influx means less neurotransmitter release, and thereby inducing less of an effect on the postsynaptic membrane. The advantage of presynaptic facilitation and inhibition is in that they selectively influence single synapses rather than the entire neuron. So what do I mean by that? Well, let's take a look. In this diagram, we're reminded that postsynaptic inhibition of a neuron occurs because of the hyperpolarization produced by the action of an inhibitory neurotransmitter on a neuron. So for that reason, inhibitory postsynaptic potentials, or IPSPs, affect the total activity of a neuron. It causes all the terminal boutons of this postsynaptic cell to release less neurotransmitter. So in contrast to that, in presynaptic inhibition, the amount of excitatory neurotransmitter released by a specific synaptic bouton is reduced, resulting in less excitation at only that specific postsynaptic neuron without affecting the other synapses. Now, convergence and divergence. This isn't a new concept. As we've already seen, communication between neurons is not always a one-to-one event. Instead, we see the axon of a presynaptic neuron branching and its collaterals or its branches synapsing on multiple target neurons. So this sort of pathway is called divergence, a divergent pathway. Okay. On the other hand, when we see a group of presynaptic neurons provide input to a smaller number of postsynaptic neurons, this forms a convergent pathway. And the combination of convergence and divergence in our neural pathway is part of what makes our nervous system so complex.